Thank you very much, everybody, for hanging in there, and thank you to the HFSP folk for their support. It's allowed us to pursue a collaboration that we've been trying for several years, and we're really grateful for that support and hopeful that our efforts are going to be working out very well. Because this is such an interdisciplinary crowd, I thought that I would start by introducing myself. I'm an evolutionary ecologist that is largely interested in understanding how environmental oscillations and environmental extremes, how organisms adapt to to that phenomena and how they cope with it. And because in many situations, animals do that by altering their behavior, over the years I've been increasingly interested in understanding how brains and cognition influence these processes. And in particular, I've been interested in understanding how changes in the environment or how the ecological forces shape the evolution of the brain, and how in turn how the evolution of the brain alters the subsequent ecology and evolution of the lineages I study. So basically the ecoevolutionary dynamics of the brain. Now, my team and I have used birds as the study system for these kinds of questions, primarily because there's a lot of really well-characterized species. We know a lot about many of them. There's almost 11,000 species of birds living in the world right now. They have a wide range of cognitive ability, so that idea that you might have heard of bird brains as somehow a derogatory term, it's a misconception because birds have a much higher number or density of neurons per volume of brain. So there's a wide range, and even some of the species tend to have cognitive abilities that rival those of primates, so things like crows and parrots, for example. Now, the interesting thing is that they also have a hyperdiverse ecology, hyperdiverse morphology, and natural history, allowing us to pursue these questions that are that sometimes are really difficult to test with small effects. But perhaps one of the most important reasons why we study birds in this case is that they have a convenient brain evolution. And what I mean by that is that most of the volumetric changes in evolutionary history of this clade have been correlated with specific changes in the telencephalon, and particularly in areas of the pallium that are involved with the kinds of traits that we think about when we talk about intelligence and complex cognition. And using that approach, therefore, we've been able to use brain size as a proxy for cognitive abilities, and we've discovered a range of interesting things about the evolution of cognition, the role of brains in ecology and different things. And here I'm just showing a small sampling of papers from my lab. But if you really think about it, at the end of the day, all of these studies are still somewhat frustrating because even though they tell us that the brain is involved in all these processes, at the end of the day, I mean, it's still a big black box, and we don't know what is it about the brain that changed, that created those associations. And because of that reason, I've spent over a decade trying to find data that we could use to inform or to get a better idea about the mechanistic underpinnings of all these relationships. And in that search, one of the papers that really struck my eye was by my now co-PI Onur Turkin's group, in which he painstakingly proceeded to map the connections of every single neuron in the pigeon's telencephalon. Now, the reason I was so excited about this particular paper was because I started thinking, like, if we could somehow map this wiring diagrams of all the different brains, we could potentially do comparative analysis of understanding the differences in network structure that yield those differences in cognitive ability. So some species may be more connected, may have more redundant connections, may have small world structures, those kinds of metrics that we get typically from graph analysis and network theory. Now, obviously, we cannot do this with Onur's technique because it took him and a group of about 8 people over 5 years to map just one single species and not even the entire brain. But at the time, I started thinking, like, well, we've had a lot of recent advances in terms of brain imaging, particularly in the field of fMRI. And then I thought, like, oh, maybe we can use this particular field to try to actually do the functional connectivity of all these different species and attempt kind of like an analysis with a much broader depth and much more mechanistic understanding than anything that we have been able to do so far. Unfortunately, reality check gave me a big, swift kick in the butt, and it made me realize that fMRI was actually really complicated because it's a very expensive machine to purchase and to operate. It is non-portable, meaning that I have to bring all the animals, or that I can only work with animals that I can bring to the lab. And most importantly, it is very sensitive to movement, meaning that in order to image these animals, I have to spend weeks, sometimes months, training them to be calm and collected in the imaging scanning condition, sometimes for periods of about half an hour. That is actually almost not possible with most species. So most of the time what people do is that they sedate animals, which in the end ends up disrupting the very patterns of functional connectivity that I want to study. So basically fMRI is just not the way to do it. Now, the goal of our HFSP project is to actually come up with an imaging alternative for mapping the functional connectivity of avian brains at large scales at a fraction of the cost in the field and in the lab, and most importantly, without the need for sedation. Now, if you guys have ever played with a flashlight, you can see like with this hand that if you have a strong enough source, the light can actually penetrate tissue quite deeply. So my second co-PI, Joseph Kolber, has been using this kind of technique to create scans of the human brain with comparable resolution to fMRI. And to give you a little bit of a sense of how this technique works, it basically works by illuminating the brain. So it has coupled detectors and sources, and we illuminate the brain with 3 different kinds of wavelengths that we later kind of like, using our knowledge of how light diffuses through the tissue, then what we get in the receptors, we use it to reconstruct basically a specific kind of color. And the principle behind optical tomography, and actually all the optical methods, is very similar to the one we use in fMRI, which is that whenever a neuron fires, there's a coupled pulse of blood that is provided by the neurovasculature around the neuron to provide the energy and the oxygen needed. So by looking at the brain for a prolonged period of time, you're able to tell where and when the brain is being active by looking at basically the pulses of blood where they're occurring. Now, the second principle is that when 2 neurons are connected, if one of them spontaneously fires, the second one also, or the one that is connected to it, also tends to fire. Which means that if you look at correlated regions, or regions that are highly connected, like in this case the one highlighted by red and orange dots, then you find that the patterns of activity are very similar over time, displayed here in the time trace to the right. And those patterns actually are quite distinct to the areas that are not connected to those. So in this particular case, the blue area, so you can see that the blue trace is very different from the red and the orange. So the next step is to assess basically the correlation between different portions of the field of view, showed here in that heat map, in which red values relate to like higher correlation. Now, of course, the sensitivity or the resolution of our device is not necessarily the same as the functional arrangement of the part of the brain that we're looking at. So the first step in the process involves then to realize which parts of the brain are actually, oh, all right, well, I'll keep like freeloading here. So it involves figuring which parts of the brain actually are acting as a single unit. So what you should be seeing here is kind of like this brain parcellated or broken down into little different pieces that basically relate to what are the functional units within the brain. Oh yeah, great. So these are the parcels that we get. And once we have that, we can actually use the average activity in those parcels to figure out how they're connected and to ultimately create these functional connectivity networks that we're interested in studying. All right, so the objectives of this particular HFSP project, we're just beginning in our first year, but we divided into 3 different things that we want to do. In the first one, we're going to use a very simple technique that is it's a precursor of tomography, but it only uses 1 source and 1 detector. And the reason why we do that is to basically optimize all the different stages of this process that I just told you. After that, what we're beginning to do now is to create the actual optical tomography device that allows us to do these 3D imaging scans on birds. It involves basically a helmet and a whole bunch of different sources and receptors or detectors. And the final objective is to actually use this device to image the brains of 4 species with dramatically different cognitive abilities, so that we can start asking the question of how does network structure lead to different cognitive function, and we can start kind of like demonstrating the feasibility of our approach for large-scale, truly large-scale comparative analysis of cognition and studies on the evolutionary dynamics of intelligence. Now, in our first year, we've been able to complete entirely this first objective, and I just want to kind of like express kind of like that what our objectives were in this particular study. If you want to show that this technique works, the very first thing we need to show is that the imaging that we're producing really is able to recover the kinds of structures that we already know from anatomical and functional studies that exist, that is able to recover some of the behavior that we know that exists in the brain, and that the images that we're producing are largely consistent within individuals over time and across individuals. So they're basically seeing the same things when we do this multiple times. And finally, and probably the most important thing, is that we need to demonstrate that our approach is as good, hopefully better, than fMRI, which is the current gold standard in this field. So here are some results. The region of interest that we use for our proof of study, our proof of concept study, was in the superficial area of the bird brain, where we find the avian woolst, which is involved in processing somatosensory stimuli, and it's divided into a visual portion and a somatosensory part. But from this particular point of view, we should also be able to see parts of the hippocampus and parts of the meso and the nidopallium. And what we find is that when we image multiple different individuals and we reconstruct the parcels that we were talking about, we largely recover the same anatomical areas that we're expecting. So there are, of course, some differences between individuals, but for the most part, we get exactly what we were expecting to see. And most importantly, and actually to our great pleasure, we find that when we do the same kind of analysis using fMRI data, we basically get the exact same result. So like first objective, kind of like checked out. The second thing is that when we look at individuals over time, we find that we reconstruct the same kinds of structures. So in this particular case, I'm showing the results from an imaging session of 5 minutes in the left and an imaging session of another 5 minutes that were taken later. And what we find is that in both cases, we reconstruct largely the same kinds of structures. Now, you may see that there are some basic differences, and those are expected because as the individuals wander, think about different kinds of things, the structural arrangement or the functional arrangement slightly changes. But overall, the general patterns, the recovery of the avian woolst, the hippocampus, and the mesonidopallium remain. Finally, when we try to assess the connectivity and try to assess whether we find the same kinds of patterns that we expect from what we know that is happening in the pigeon, we thought that perhaps one of the good things to look at would be the connections between left and right hemisphere, which we know that should occur. And to do that, we take those heat maps that I was showing you before, and we arrange them by separating them by the regions, by the parcels that we're talking about, and by hemisphere, such that on the right we have the exact same data but organized in a slightly different way, so that we have on the top left the connections between just the left hemisphere. On the bottom right we see the connections between just the right hemisphere, and the off diagonals we see the connections between left and right. And what you find here is that not only, oh sorry, I don't know how to get back, but to the punchline. Not only do you find that these units come on exactly as you expected, so the regions are kind of like clear, like the parcels are clear, but also that the colors on one hemisphere are strongly correlated with the same colors in the other. So these homotopic relationships, these correlations between left and right, are pretty evident. And more importantly, we find that it's exactly the same pattern that we observe when we look at fMRI. And you might see that the colors of ours are more intense, is basically because our method has a higher resolution, but that's not necessarily that it's going to be that forever. It's because our wide field camera right now is a high resolution camera. But in the end, it's basically the same result. So in conclusion, we find that the optical methods that we're using kind of like really allow us to recover what we want. They show strong concordance between what we know about anatomy and functional connectivity. They give us consistent results over time and across individuals, and they provide similar outcomes, or as you saw, slightly better outcomes than fMRI. But now we actually can start doing the really exciting stuff, which is to move on to the 3D imaging. Right now, our current prototype, we built multiple different versions of this. It involves basically this gnarly looking helmet with many different sources and detectors. Our current prototype involves 16 sources and 21 detectors. And this is kind of like just an example of the point spread simulations that we do to understand how light should be diffusing through the tissue, and then we use that to create all the models to understand what we're getting. And even though we already have had good imaging sessions for at least a couple individuals, we're not quite ready that for prime time, but we're about to be, and we should be able to share that with you pretty soon. So I want to end up, I think, right now by acknowledging, of course, the incredible help from my co-PIs. Joseph Culver is the expert in imaging and radiology. Onur Günterkin is our avian neurobiology expert, and now we've added through the wonderful new program, we've added a colleague that is working on AI and image analysis. And finally, I would really like to acknowledge Dr. Katie Chenard, which is a postdoc funded by HFSP, who's been helping us put all of this together, and the other members of our team. And I hope to take some time for questions. Thank you very much.